In 2014, a team of researchers at the University of Queensland built what they believed was a perfect color vision test for the mantis shrimp. The mantis shrimp has 16 types of photoreceptors in its eyes. Humans have 3. Honeybees, which are considered to have exceptional color vision among insects, have 4. The mantis shrimp, with its 16 channels, was widely assumed to possess the most sophisticated color perception of any animal on Earth, capable of seeing colors that humans cannot imagine, operating in a visual dimension so far beyond human experience that comparing the two seemed almost meaningless. Science magazines had been writing about this for years. The mantis shrimp's vision had been described as superhuman, as seeing the rainbow behind the rainbow, as experiencing colors the way a satellite sensor experiences the electromagnetic spectrum. The Queensland team designed a discrimination test to measure just how precise this color vision was. They trained mantis shrimp to associate specific wavelengths of light with a food reward, then presented them with pairs of colored lights at progressively closer wavelengths to find the threshold below, which the animals could not tell the difference. The results came back. The researchers looked at them, then they looked at them again. The mantis shrimp could discriminate between wavelengths approximately 25 nanometers apart. A human can discriminate wavelengths as little as 1 to 4 nanometers apart. The mantis shrimp, with 16 photoreceptors, was 6 to 25 times worse at distinguishing color than a human with 3. The paper was published in Science. The title was careful and measured. The scientific community's reaction was something closer to bewilderment, and the question it opened: why would an animal evolve the most complex visual hardware in the animal kingdom and then use it to perform worse than a mammal with a fraction of the equipment? Is one of the most genuinely strange puzzles in modern biology. To understand the paradox, you first need to understand how human color vision actually works, because the way we see color is not what most people assume. Your eye contains 3 types of cone photoreceptors, each sensitive to a different range of wavelengths. One peaks in the shortwave range, roughly 420 nanometers, perceived as blue-violet. One in the medium wave range, roughly 530 nanometers, perceived as green, and one in the long wave range, roughly 560 nanometers, perceived as yellow-red. No individual cone tells you what color something is. What the brain receives is 3 numbers, the relative activation levels of all 3 cone types, and the color you perceive is computed from the ratio between those numbers. This comparison process is crucial. The brain takes 2 signals and asks, how different are they? A small difference between the blue-sensitive and green-sensitive cones produces a perception somewhere in the blue-green range. A large difference, weighted differently, produces something at the red end. The precision of this comparison, the ability of the visual system to detect very small differences between cone activation levels, is what allows humans to distinguish wavelengths just 1 to 4 nanometers apart. We are not measuring color directly. We are computing it from differences between broadly overlapping sensors. The mantis shrimp does something fundamentally different. Its 16 photoreceptor types are not broadly tuned, overlapping sensors like human cones. They are narrowly tuned. Each responds strongly to a very specific, narrow range of wavelengths and barely responds to anything outside that range. Instead of 3 overlapping curves covering the visible spectrum, the mantis shrimp has 16 narrow spikes, each centered on a specific wavelength, covering the range from deep ultraviolet to far red. When the Queensland team's results showed that mantis shrimp could only discriminate wavelengths 25 nanometers apart, the reason became clear. The mantis shrimp is not comparing signals between photoreceptors. It is reading each receptor independently. A color is not computed from the ratio between channels. It is identified by which channel fires most strongly. This is not a comparison system. It is a recognition system. The animal has essentially 16 labeled bins, labeled this specific wavelength, this other specific wavelength, and so on. And it identifies colors by determining which bin the incoming light falls into most strongly. The difference in performance is the direct consequence of this architecture. A comparison system with 3 broad sensors can resolve extremely fine differences because it is sensitive to small changes in the ratio between signals. A recognition system with 16 narrow sensors is limited in precision by the spacing between those sensors, roughly 25 nanometers, because colors that fall within one bin are perceived as identical. The mantis shrimp does not see more colors than you. It sees color differently. And for its purposes, its system works extraordinarily well. It just does not work the way anyone assumed. What the recognition system is actually optimized for was the question researchers turned to next. The leading hypothesis involves speed. A comparison system, like human color vision, requires the brain to receive activation data from multiple receptor types, compute the ratios, and generate a color perception from the result. This takes time. Not much time by human experience, but measurable time at the neural level. A recognition system, where each receptor type directly signals the presence of a specific wavelength without requiring comparison to anything else, could in principle operate faster. The signal from one channel is enough to identify the color category without waiting for cross-channel comparison. Mantis shrimp are among the fastest predators in the ocean. Their striking appendages, the raptorial claws they use to overwhelm prey, accelerate to the speed of a bullet and deliver forces equivalent to roughly 1,500 newtons on impact. The entire strike takes place in approximately 2 to 3 milliseconds. At this timescale, the animal cannot afford slow visual processing. A color recognition system that identifies this is the target color in one neural step, rather than through a cross-channel computation, may be precisely what a predator operating at these speeds requires. The mantis shrimp does not need to distinguish between 2 similar shades of orange. It needs to identify, instantly, whether the thing in front of it matches the color signature of prey, of a rival, or of a potential mate. And it needs to do this fast enough to act on the information within a time frame measured in milliseconds. 12 to 16 labeled color channels, each optimized for rapid recognition rather than precise discrimination, may be a better solution to that specific problem than 3 broadly overlapping channels that require neural computation to resolve. This is an important point. The mantis shrimp's visual system is not worse than human color vision. It is different, built to solve a different problem under different constraints. The Queensland experiment revealed that it cannot do what human color vision does. It did not reveal that it cannot do what mantis shrimp color vision needs to do. The photoreceptors in the mantis shrimp eye are not arranged randomly. They are organized into a horizontal band called the midband, a specialized region running across the center of each compound eye that contains the 12 to 16 spectral receptor types. Above and below the midband are more conventional compound eye regions used for motion detection and spatial resolution. The midband does not provide a panoramic field of view. It is a narrow scanning strip. To build a complete picture of the color composition of a scene, the mantis shrimp must scan its eyes across the visual field, moving the midband over different parts of the scene the way you might move a barcode scanner across a product. The color information is not gathered simultaneously from the whole visual field. It is gathered serially, point by point, as the eyes move. This scanning behavior has been directly observed. Mantis shrimp examining novel objects make characteristic rapid eye movements, raster-like scanning patterns that sweep the midband across the object systematically. They are not looking at the object in the way you look at it. They are reading it line by line, with a spectral sensor that records which color categories are present where. The result is a color map of the object, not a smooth perception of continuous color gradation, but a categorical inventory. This region is in wavelength bin 7. That region is in bin 11. This edge marks a transition between bins 3 and 8. A predator that hunts camouflaged prey on complex reef backgrounds could potentially use this categorical map to detect subtle deviations in color category that would be invisible to an eye resolving color smoothly. A prey animal that is almost perfectly camouflaged may still show color category boundaries that its background does not, and the mantis shrimp system, with its narrow, independent color channels, might detect those boundaries even where a comparison-based system would smooth them away. This is still hypothesis. The neural pathways downstream of the midband receptors have not been fully mapped. What the mantis shrimp's brain does with the spectral information it receives from the midband scan is not fully understood. The 2014 paper that revealed the discrimination paradox did not close the question. It opened several new ones that researchers are still actively investigating. There is one more feature of the mantis shrimp visual system that gets significantly less attention than the photoreceptor count, possibly because it is harder to explain in a sentence. Mantis shrimp can detect the polarization of light. Not just linear polarization, the orientation of light's oscillation, but circular polarization, which refers to light whose polarization rotates helically as it propagates. Circular polarization detection is essentially unknown in the animal kingdom outside of stomatopods. It has been found in a small number of other species, but mantis shrimp appear to have evolved it independently, for purposes that have only recently been understood. Mantis shrimp communicate using circularly polarized signals. Specific regions of their shell reflect circularly polarized light in patterns that are invisible to animals without circular polarization sensitivity, which is nearly every other animal on the reef. Their signals are, in effect, a private channel. A mantis shrimp signaling its health, reproductive status, or territorial claims to another mantis shrimp is broadcasting in a wavelength that predators and competitors cannot see. The physical basis of this signaling system was identified in 2008 by researchers studying the reflective structures in mantis shrimp shells. The structures are multi-layer photonic reflectors, biological analogues of the thin film optical coatings used in camera lenses and LCD screens, arranged in a specific orientation that produces circularly polarized reflection. The animal grew its own optical filter at a microscopic scale through biological processes that produce structures functionally equivalent to precision optical engineering. The mantis shrimp uses its circular polarization detectors to read these signals. The spectral photoreceptors in the midband handle color recognition. Separate photoreceptors handle linear and circular polarization. The compound eye regions handle motion and spatial resolution. The mantis shrimp does not have one visual system. It has several overlapping but functionally distinct systems operating in the same eye simultaneously, each handling a different kind of visual information independently. The broader question the mantis shrimp raises, the one that the Queensland researchers and the neuroscientists who followed them have returned to repeatedly, is what the relationship between sensory hardware and sensory experience actually is. The implicit assumption in most popular coverage of animal vision is that more photoreceptors means richer perception, more channels, more colors, more experienced. The mantis shrimp was the extreme case of this assumption. 16 channels, therefore 16 times the color experience of humans. The 2014 experiment demonstrated that this is not how it works. More input channels do not automatically produce more refined perception. What determines perceptual resolution is not the number of channels, but what the brain does with them, how the signals are processed, compared, combined, and interpreted. The mantis shrimp took 16 channels and built a rapid recognition system that categorizes color rather than computing it. Humans took 3 channels and built a comparison system that discriminates color with extraordinary precision. The two systems produce radically different perceptual outcomes from the same physical input, light with a spectrum. Neither is better. They solve different problems. What the mantis shrimp actually experiences, whether its visual world has any phenomenal quality, whether there is something it is like to be a mantis shrimp scanning a reef with its midband, is a question biology cannot currently answer. We can measure what its photoreceptors respond to. We can observe how it behaves. We cannot access the subjective experience, if any, that underlies the behavior. What we can say is that the mantis shrimp's visual system is not what anyone thought it was before 2014. It is not superhuman in the way that phrase was being used. It is not a richer version of human color vision, scaled up. It is something structurally different, a recognition architecture rather than a comparison architecture, operating at speeds that match its predatory lifestyle, reading the reef in a language of color categories that no other animal we know of is fluent in. The most complex visual hardware in the animal kingdom, producing a form of perception that is, in some respects, less capable than what you are doing right now without thinking about it, and in other respects, doing things that no eye like yours ever could. If you want to see the animal whose visual system solves the same problems in the opposite direction, almost no photoreceptors, almost no light, but navigating with perfect precision at depths where vision should be impossible, that video is here.